Optogenetics, how light can change brain function. Optogenetics is a relatively new research technique. Piecing together bits of biochemistry, microbiology, and genetics, it allows precise control of specific brain circuits on demand using light. Here, you can see a cable running into the skull of a mouse. When a light at the end of the cable is activated, the mouse switches into attack mode. So what's happening? If you safely drill a hole into the skull of your average mouse and shine a light inside, turning the light on and off isn't going to change the mouse's behavior. This mouse has been genetically modified so that the neurons instigating an aggressive response have a unique type of ion channel known as a channel rhodopsin. Like typical ion channels, channel rhodopsins will let ions flow through the cell membrane when they open. Depending on whether the channel allows negative or positive ions into the neuron, it will either increase or decrease the neuron's membrane potential. Letting in negative chloride ions will hyperpolarize the cell and make it harder for an action potential to occur. Letting in positive sodium ions will depolarize the cell and make an action potential easier to reach. Unlike typical ion channels, channel rhodopsins have a light-sensitive component which controls when they open. This component is sensitive to a specific wavelength of light, which varies from channel to channel. This means that a neuron expressing more than one channel rhodopsin can react differently to different wavelengths of light. Alright, well, where do channel rhodopsins come from, and how do we get them to show up in a mouse's brain? Well, let's start with where they come from. The existence of rhodopsins, photoreceptor proteins, has been known for over a century. Our eyes wouldn't work without them. But about 30 years ago, researchers discovered a protein complex in single-celled green algae that utilizes a rhodopsin to control the gating mechanism of an ion channel. When exposed to light, cations would flow in through the channel, and flagella on the surface of the algae would begin to move the cell toward the source of light. Now, these findings were largely ignored at the time. Why would knowing why algae move in the presence of light be relevant to other fields? Decades passed, during which time other types of channel rhodopsins were found in unicellular organisms from algae to archaebacteria. It wasn't until the advent of methods allowing animals to express foreign genes that these channels would be used as a research tool. In 2005, researchers began looking into transferring the channel rhodopsin genes into other organisms. Several techniques have been used to get organisms to express them, such as splicing the gene into an embryo and breeding lines of transgenic mice, or using a virus to infect desired cells with the proper genes. Either way, the target cells need to express the channel rhodopsin protein complex, or they won't react to light. Once you've got a mouse with the right channel in the right place, a light source needs to be inserted into the brain so that the channels can actually be activated. This can be done either with a cord strung from the ceiling of the cage into the mouse's brain, or by attaching a wireless system to the inserted light source to allow more freedom of movement. Activating the light will either excite or inhibit an action potential, depending on the ions the channel lets through. This allows researchers to pinpoint specific neurons or neural circuits that control behaviors from aggression to eating. Because you can pick and choose specific circuits to activate, optogenetics holds some promise as a method of treating brain disorders. If a circuit isn't activating or deactivating when it's supposed to, optogenetics can provide the switch to turn it on or off. Deep brain stimulation, an experimental technique used for treating central nervous system disorders like Parkinson's, most often uses electrodes to activate specific regions of the brain. The electrodes activate neurons that alleviate the symptoms of the disorder. In the case of Parkinson's, they're inserted into the subthalamic nucleus. The electrodes activate neurons in the dopamine pathway that don't function normally in Parkinson's patients. As you can see in this Parkinson's patient, when the electrodes are activated, they alleviate his symptoms almost immediately. Unfortunately, electrical stimulation isn't particularly precise. It can activate regions adjacent to the target area. This can activate or inhibit other circuits, some of which might accidentally impact the activity of the target circuit. Because it lacks precision, electrical deep brain stimulation can have side effects like confusion and seizures. This is where optogenetics comes in. Light will only activate cells that actually express the channel rhodopsin. The gene for the channel rhodopsin can be tailored so that it's only expressed by the desired cells. This limits the pathways affected in a way that electrical stimulation just can't. It doesn't matter if light touches non-target neurons because it won't affect them. This means deep brain stimulation would be more effective and cut back on negative side effects.